Dear students, in today's lecture we will discuss digestion of proteins, another important topic for BSc final exam topic. प्यारे बच्चों, this is also a common topic for all competitive exams. Digestion of proteins. In our last lecture we have covered digestion of carbohydrates, means this is another topic for final exam. BSc students, note down. प्यारे बच्चों, ये जितने भी हमने नोट्स बनाए उन सब की पीडीएफ फाइल वीडियो लेक्चर उसके डिस्क्रिप्शन में अपलोड कर दिए, सभी बच्चे अपने नोट्स डाउनलोड कर लें. डियर स्टूडेंट्स, इन टुडेज लेक्चर वी विल कवर digestion of proteins. As we know, proteins are complex, large organic molecules, and these proteins are polymers of amino acids. It means in proteins, in polypeptide chains, amino acids are interlinked by peptide bonds, and these bonds are hydrolyzed by proteolytic enzymes, and this group of enzymes are named proteases as they hydrolyze the peptide bonds. So these are peptidases. Dear students, digestion of proteins starts in stomach and it completes in small intestine. As in case of man, our saliva, in our oral cavity, buccal cavity, saliva doesn't contain any enzyme for digestion of proteins. हमने कहा बच्चों, digestion of proteins starts in stomach. First of all, we will discuss digestion of proteins in stomach. In our stomach, both mechanical and chemical breakdown of food take place. Mechanical breakdown means powerful muscular movements of stomach, the churning movements of stomach causes proper mixing of food with HCl, hydrochloric acid, that is produced by the gastric glands, oxyntic cells. In gastric juice, two types of enzymes which are proenzymes, pepsinogen and prorenin. And dear students, these proenzymes are produced by peptic cell chief cells. It means in case of gastric juice, mucus is present, HCl is present, and that HCl is produced by oxyntic cell and two proenzymes. These are propepsin and prorenin. These proenzymes and HCl play important roles. First of all, role of HCl. And this hydrochloric acid which is produced by oxyntic cells of gastric glands that kills the harmful bacteria, pathogens, microorganisms present in our food. This HCl, it stops the action of salivary amylase. In our last lecture we have discussed salivary amylase remains functional in our pharynx, in our food pipe, as well as in stomach up to 30 minutes, and after 30 minutes it gets destroyed by HCl. It means the important role of this HCl that stops the functioning of salivary amylase. And this HCl, as in our gastric juice the enzymes are produced for the digestion of proteins, remain inactive. These are proenzymes. These are pepsinogen, and this proenzyme, this inactive enzyme is converted into active form, pepsinogen into pepsin and prorenin into renin by HCl. It means HCl activates these inactive forms of enzymes. HCl that provides optimum pH for these enzymes, and the specific pH which is maintained by HCl is 1.2 to 1.8. Dear students, note down all important points. The specific pH, the optimum pH on which these enzymes remain functional. They show maximum activity, that is 1.2 to 1.8 pH. And this HCl, it causes softening of food, as well as the HCl dissolves the cementing material present between the adjacent cells. Very important, NEET students, competitive exam students, it is believed that the HCl regulates, it controls the opening and closing of pyloric sphincter. It means the acidic food from this stomach enters in small intestine through a sphincter, that is pyloric sphincter, and its working means its opening and closing is regulated by HCl. Note down all important points. Role of pepsinogen. Pepsinogen, that is proenzyme, that is inactive enzyme, and this enzyme in presence of HCl or in acidic medium, it gets activated. In acidic medium, it becomes most active at specific pH. At that pH is 1.2 to 1.8 pH. And this pepsinogen, inactive enzyme, in presence of this HCl at specific pH, it becomes pepsin. This is principal proteolytic enzyme of gastric juice, the main enzyme, principal enzyme of gastric juice, that is pepsin. And this active form of pepsinogen, that is pepsin, this pepsin acts on proteins and converts these protein molecules into peptones plus proteoses. These are fragments of proteins. Role of prorenin. In our gastric juice, two proenzymes are present. One is pepsinogen, and that pepsinogen in presence of HCl becomes active, that is pepsin, and that pepsin converts the protein molecules into peptones and proteoses. Next, second enzyme, very important for all competitive exams, that is prorenin. Prorenin, it means it is zymogen. It is proenzyme. It is inactive form. And this inactive form, in presence of this HCl, at specific pH, the optimum pH, that is 1.2 to 1.8, and this prorenin becomes active, and the active form is renin, or it is competition student, it is rennet, or it is chymosin. This active form of prorenin, which is renin, it acts upon milk protein, and the milk protein is casein, and this casein in presence of this renin, active form of this prorenin, converts it into paracasein, and this paracasein in presence of calcium salts, it becomes calcium paracaseinate, and this process is conversion of this milk protein casein into calcium paracaseinate. It is named curdling of milk. Dear students, this is coagulation of milk. This is clotting of milk, not clotting of blood. This is clotting, coagulation of milk. Milk protein gets coagulated. This process is very important, curdling. Now, on this calcium paracaseinate, on this coagulated milk, this active form, pepsin, it acts, and this pepsin converts this coagulated milk, calcium paracaseinate, into peptones and proteoses. Again, these are fragments of proteins. After partial digestion of food in stomach, this chyme, the highly acidic food, and this acidic food, it is chyme. The food is passed, the chyme is passed in small intestine. In small intestine, this chyme, this acidic food meets with 3 juice. Dear students, 3 types of juice. One is bile juice and the pH is 8.0. And the bile juice is produced by liver. And second juice, that is pancreatic juice. Its pH is maximum, that is 8.8, produced by pancreatic glands. And third type of juice, very important, that is small intestinal juice. Intestinal juice, produced by the glands of small intestine. Its pH is 8.3. Each and every point very important. Note down, all competition students. It means the chyme, the acidic food, enters in small intestine. And in small intestine, 3 types of juice. First of all, role of bile. The liver juice, the bile, that doesn't play any role, as in this bile. No enzyme for digestion of proteins. Role of pancreatic juice. This pancreatic juice, it contains 3 proenzymes. 3 proenzymes means 3 protein digestive enzymes are found in inactive form. These are trypsinogen, chymotrypsinogen, and procarboxypeptidase. In pancreatic juice, 3 protein digestive enzymes found in inactive forms, in proenzyme forms. This trypsinogen, this inactive form, this proenzyme in presence of enterokinase, enteropeptidase, and this this special enzyme, enterokinase, it comes from Saccus entericus, small intestinal juice, and this enterokinase, it is known as very famous enzyme for all competitive exams. This is an activator enzyme. This enzyme activates this trypsinogen into an active form, which is trypsin. And this trypsin, it is an autocatalysis process. Once it gets catalyzed, it becomes activated. It activates other enzymes like chymotrypsinogen. This inactive form in presence of this activated trypsin converts into chymotrypsin. And this procarboxypeptidase, another inactive form, in presence of this trypsin activated form, it becomes active, that is carboxypeptidase. Now, proteins in presence of trypsin converts into large peptides. Proteins in presence of chymotrypsin enzyme, and these proteins are converted into large peptides. Another very important protein digestive enzyme is also present in pancreatic juice. 3 proenzymes and 1 enzyme, that is elastase. And this elastase enzyme converts the elastin proteins into large peptides. And on these large peptides, carboxypeptidase acts and converts the large peptides into dipeptides and amino acids. Dear students, the last important point of this, role of intestinal juice in proteins digestion. And this intestinal juice, which is Saccus entericus, that contains 3 enzymes. हमने कहा 3 enzymes for protein digestion. One is enterokinase, and that involves in the activation of this trypsinogen of pancreatic juice, and 2 more enzymes. These are aminopeptidase and dipeptidases. Large peptides of pancreatic juice, and these large peptides in presence of aminopeptidase converted into dipeptides and amino acids. And the dipeptide is last enzyme which acts upon these dipeptides to convert into smaller units. These are amino acids. It means the complex large protein molecules are hydrolyzed. The peptide bonds are hydrolyzed. These are simplified. And the simple forms are amino acids. It means the digestion of proteins that was started in stomach and that completes in small intestine. Dear students, we have covered two very important topics, digestion of carbohydrates, and today's lecture we have covered digestion of proteins, and tomorrow we will discuss digestion of fats. Thank you.